Hello, everybody, and thank you for the opportunity to be involved in the CJ BIO ANH conference this afternoon. The title of my presentation today is to examine the various impacts of dietary fibre on nutrition and the physiology of monogastric animals, being in pigs and chickens. And as an outline of my presentation today, I wish to cover four major areas. First, an overview and definitions of dietary fibre in the context of monogastric animals, some physiological and microbiological effects of dietary fibre, some of the nutritional and production impacts of dietary fibre, then to move on to looking at some functional fibre fractions and their impacts, and to make some general conclusions. In commercial pork and poultry production, plant carbohydrates represent the main fraction of a diet, and indeed they account for more than 66% of total dry matter and comprise 60 to 70% of total energy intake. However, a fraction of that dietary carbohydrate is not digested by the enzymes in the small intestine of pigs and chickens, and becomes a substrate for bacterial fermentation in the large intestine of the pig or the ceca of birds. This is called dietary fibre, and it has well-known physiological, microbiological, immunological and nutritional impacts along the gastrointestinal tract in pigs and poultry of all growth and breeding stages. These impacts are generally regarded as being anti-nutritive. For example, causing an increase in viscosity, reducing overall energy and nutrient digestibility and availability, and resulting in suboptimal performance. And that really underscores the importance of enzymes and enzyme application, which is a key part of this conference. But more recently, there's growing interest in the use of dietary fibre in pig and chicken diets, not only to reduce feed costs, but also their beneficial effect on gut health, the impact of functional fibre, as well as on welfare and environmental benefits. One of the key understandings with dietary fibre is the classification. And in general, dietary fibre can be classified according to the location within the plant cell, the various analytical methods of determination, such as soluble, insoluble dietary fibre, total dietary fibre, non-starch polysaccharides plus lignin, the Van Soest methods, and the incorporation of resistant starch. And then there are nutritional and physiological definitions. Now, all of these definitions are interrelated to impact on the animal, but they're often measured separately, and there is no connection between them to examine their impacts on pigs and chickens. But this is a diagram which basically describes the different chemical methods of analysis and outcomes for plant carbohydrates. On the left-hand side, we have the cell contents. These are the water-soluble and non-structural carbohydrates, such as starch, oligosaccharides, and resistant starch. On the right-hand side are the cell wall components, or the cell wall carbohydrates. And these are associated with compounds including beta-glucans, and then the traditional terms, hemicellulose, cellulose, and also lignin. Now, it's the cell wall components that comprise the major proportion of dietary fibre, and there's different ways that they can be analysed. For example, the Van Soest analysis talks about, for example, NDF, ADF, and crude fibre, which describes aspects of the indigestible fraction of the cell wall. A relatively new term, 20 to 25 years ago, now called non-starch polysaccharides, which splits these cell wall components into soluble dietary fibre and insoluble dietary fibre, and together they become total dietary fibre. But then more recently, there's a move towards trying to assess more physiological definitions of plant carbohydrates, from the less fermentable fractions right through the spectrum to the more fermentable fractions. And this is where dietary fibres based on fermentability, water holding capacity, and bulk density can have relevance. Here are some different fibre sources, such as sugar beet pulp, soybean hulls, wheat bran, oat hulls, and resistant starch. And what this table does is describe the fermentability, water holding capacity, and bulk density of these fractions. For example, if we look at fermentability, The greater the number of these signs indicates the higher extent of fermentability and water holding capacity compared to other ingredients. Sugar beet pulp, for example, is quite fermentable and attracts a lot of water compared to oat hulls, which are less digestible and attract less water. But these are also more dense, which comes into consideration for diet formulation. But this is a different way that we can look at classifying dietary fibres. We can talk about this a lot more, but in general, what we see is that the physiological properties and fermentability of fibrous ingredients are difficult to predict from chemical composition in general. The properties and fermentability are more related to their solubility, viscosity, physical structure, such as particle size, and water holding properties. And indeed, it has been shown that about 90% of the variation in the energy value of fibres is related to their variation in fermentability. It's very important. Therefore, the effective use of fibrous ingredients depends to a large extent on the adequate estimation of the physico-chemical properties and nutritional value of the different ingredients. Now, it's quite apparent that some of the current and relatively quick and cost-effective methods of fibre analysis, such as the traditional fibre outcomes like crude fibre, do not necessarily relate to their functional value to the pig and the bird. And therefore, robust and relevant methods for the determination of the physiological functionality of dietary fibre sources still needs to be explored. And I'll cover some of that later. So let's move along and talk about some of the physiological and microbiological effects of dietary fibre. Well, it's known that hindgut fermenters, such as pigs, generally attempt to increase feed intake when offered a higher fibre diet. And in fact, it's been known for more than 50 years that for every 1% increase in crude fibre content up to 10% in the diet, or 100 grams per kilogram, a pig will increase its intake by 3% to maintain a constant level of digestible energy intake. However, a threshold point is reached at some point where higher fibre contents limit intake, but the pigs cannot and chickens cannot eat anymore because of the bulking effect. Increasing the dietary fibre content will generally decrease digestive retention time, that is relative to a very low fibre diet, with transit through the gastrointestinal tract being largely a function of retention time in the large intestine. This in turn is related to fecal bulking capacity. Fibre particle size is also known to be inversely related to retention time, but in pigs, at least in growing pigs, it's the insoluble fraction that largely impacts retention time. And this was a study that was conducted to look at 3 levels of total dietary fibre, classified as low, medium, or high. And it looked at the mean retention time. This is shown on the y-axis in each of these graphs, in the stomach, the small intestine, and the large intestine. In the stomach, we see no impact of the fibre content, and this is the insoluble fibre content between the different diets in terms of retention time. And this is split into the solid phase in the blue bars and the soluble phase in the grey bars. No impact of fibre content in the stomach, but in the small intestine, and particularly the large intestine, what we see is a decrease in mean retention time with an increasing level of total dietary fibre, as in soluble fibre content. And this is particularly pronounced in the large intestine. We see significant reductions in transit time, particularly in the soluble phase, as the fibre content is increased. And this has important impacts in the post-weaning period. This is some work which looks at the crude fibre content in diets from 0.5% to 5.5%. It looks at transit time across the whole intestinal tract in hours against the number of days after weaning. You can see very clearly that there are 2 things. First is that there is a decrease in transit time as time after weaning progresses. And secondly, that as the crude fibre content increases, the transit time decreases. And this has important consequences for the post-weaning phase because it's known that the combined effects of stress extend transit time and can increase the resident time of substrates and bacteria. Moving along, some of the other general physiological consequences include the increased production of saliva. gastric juice, hydrochloric acid and pepsin in the stomach, as well as increased pancreatic juice flow and production and stimulation of biliary secretions from the liver. Dietary fibres can also modify profoundly the secretion of various gastrointestinal hormones, such as glucagon-like peptide 1, cholecystokinin, and secretion, that have important impacts on metabolism and feed intake. And the ileal brake, caused by secretion of GLP-1, is a well-known factor of including high levels of fibre in the diet. Dietary fibres have been implicated in aspects of innate immune function and barrier function of the small intestine. I will discuss some of these aspects later, and have also been shown to impact the morphology and function of the epithelium and to increase endogenous losses. And I just want to talk about one study that looks at the impact of xylanase, an added xylanase, on epithelial function in weaner pigs, just to demonstrate the associations between mucin and the impact that a xylanase can have. This was a study that looked at 2 Australian wheat types. Wheat is a common ingredient fed to young pigs. Wheat 1 and wheat 2. Now, wheat number 1, the pigs ate 26% less and grew 23% slower than pigs that were offered wheat 2. Their total NSP contents, non-starch poly contents, were similar, but wheat 1 contained more soluble non-starch polysaccharides, nearly 100% more, but less insoluble non-starch polysaccharides compared to wheat 2, and the protein and starch contents were relatively similar. Now, we know that if we take samples of wheat, there's going to be tremendous variation in the non-starch polysaccharide content. This is a study that looked at 12 English wheat samples. It looked at the total NSP content in the yellow bars and the soluble non-starch polysaccharide content in the blue bars. A 56% difference between the wheats in total NSP and a nearly 50% difference between the wheats in soluble NSP. These are profound differences in non-starch polysaccharide content that have important influences on production, microbiology, nutrition, and gut health. Yet these are often not measured, at least routinely, for inclusion in wheat diets, wheat-based diets for pigs. It also underscores the importance of the use of enzymes in wheat-based diets. But back to this particular diet, xylanase was added at 300 milligrams per kilogram, and the apparent digestibility of the sugars was assessed along the gastrointestinal tract. What this diagram shows is a relationship between fucose and the appearance of fucose in the duodenum, the jejunum, and in the feces. In this study, fucose was measured as an indicator of endogenous losses via mucus secretion. Therefore, the higher the concentration of fucose, then the greater the level of endogenous losses, which includes amino acids, of course. What we see are differences, essentially differences between the addition or the not addition of an enzyme. So the blue bar here is wheat number 1, and the red bar here is wheat number 2. We see the impact of non-starch polysaccharide content on endogenous losses, but these are essentially nullified when an enzyme is added, irrespective of the non-starch polysaccharide content. We see the same in the jejunum, and as we would expect, there's no impact in the feces. So this study really shows the importance of trying to mitigate the negative anti-nutritive effects of arabinoxylans, at least in wheat. Dietary fibre also is involved in fermentation, and we know about short-chain fatty acids. Short-chain fatty acids are produced mainly in the distal bowel, the cecum and the colon in pigs, and in the ceca in chickens, where the majority, about 90% of the short-chain fatty acids, are metabolized and absorbed. The short-chain fatty acids are primarily taken up and metabolized by the epithelial cells in the colon, called the colonocytes, and this is mainly butyrate. But short-chain fatty acids are also used as energy sources by the liver and skeletal and cardiac muscle cells. The extent of the fermentation by the bacteria, and therefore the yield of the short-chain fatty acids or volatile fatty acids, really depends on the quantity and type of dietary fibre and the age of the animal. So, for example, short-chain fatty acid can contribute up to 15% of daily maintenance energy requirements of growing pigs and up to 30% of daily maintenance energy requirements in sows. These are valuable and very energetically important contributions to the maintenance energy requirements of these pigs. And the absorption of short-chain fatty acids also facilitates, very importantly, the absorption of water, minerals such as sodium, and other plant compounds. Now, the plant polysaccharides that I've talked about previously provide this essential fermentative substrate to the microbiome. That includes the bacteria, fungi, protozoa, and archaea, but in pigs and chickens, we're predominantly talking about the bacterial microbiome. And fibre is known to impact substantially bacterial composition, diversity, and metabolic capabilities. The gastrointestinal tract of poultry and swine are highly diverse, containing over 1,000 bacterial species, belonging mainly to the phyla Firmicutes, Bacteroidetes, and the Protobacteria. And it's via the different enzyme systems in the bacteria, such as hydrolases, lyases, and esterases, that the gut-associated bacterial communities are able to break down and ferment the complex plant polysaccharides into short-chain fatty acids, and that for then to provide energy, but also to provide various gut health benefits to the animals. And again, I will talk about that in a little while. Now, there is a lot of information about fibre and fermentation and the microbiome, but I just wanted to show a study here conducted in 1987 in adult gills. And it shows, and this is in the days before advanced microbiome techniques, and it essentially shows the relationship between feeding these gilts a controlled diet or one with 40% alfalfa meal. And these two graphs look on this y-axis here, the cellulase activity, on this axis here, the cellulitic bacteria or the numbers of cellulitic bacteria against the time after introduction of feeding of the alfalfa meal. And to summarize this very quickly, we see that in response to alfalfa meal, the amount of cellulase activity significantly increases, and with it, an increase in the number of cellulitic bacteria. But the number of cellulitic bacteria decreases over time, but there is not a significant decrease in the cellulase activity because the numbers of cellulitic bacteria increase. In this particular study, the substrate availability increased the predominance of various bacteria associated with fibre fermentation, and that is a common outcome of a change in diet feeding with respect to fibre. Now to move on to some of the nutritional and production impacts of dietary fibre in pigs and birds. Well, typically pigs adapt to consuming higher fibre diets by increasing gut volume, or they increase their intake, as I mentioned before. That increases gut volume and weight, particularly the large intestine, through that increase in visceral weight, increase in muscle growth, and that will increase maintenance requirements of the organ because of the oxidative requirements of those of that epithelium. Generally speaking, carcass weight or dressing percentage is reduced at a similar body weight at increasing inclusion levels of fibre-rich co-products. So therefore, there needs to be a balance or a trade-off between feeding these animals and the economic value of the carcass. The increased viscera size and weight at an equal intake of net energy and SID amino acids may reduce protein deposition in the carcass. And back in 1998, we wanted to examine this further. We fed pigs between 20 and 55 kilograms, a number of diets that varied in soluble, insoluble, and total non-starch polysaccharide contents, and we assessed the weights of the cecum and colon, performance, and dressing percentage at slaughter. This graph shows a relationship on the y-axis between the dressing percentage of these pigs at slaughter and the daily intake of soluble dietary fibre. And we found a very nice negative relationship such that dressing percentage reduced 2.5% for each 100 gram per day increase of soluble fibre. Now, this can have profound economic impacts on the value of the carcass, and therefore needs to be considered in feeding diets for finishing pigs. There are, of course, many other fibrous-rich ingredients available, and one of those is DDGS, distillers dried grains with solubles. And this is some work from a number of years ago which looks at the impact of the time of withdrawal of feeding of DDGS before slaughter on dressing percentage. You can see very clearly that if you take out, in this case, the 30% DDGS, if you take out that from the diet 3 weeks or 6 weeks before slaughter, then the negative impacts on dressing percentage can be mitigated. A similar situation happens in birds. This was a study where 3 diets were fed: a control diet or low crude fibre content diet with 1.5% crude fibre, or 2 diets, one with 5% oat hulls or a diet with 5% sugar beet pulp. And the main focus of this study was on the gizzard. And you can see very clearly that the addition of 5% oat hulls or the addition of 5% sugar beet pulp relative to the control diet increased the empty weight of the gizzard, increased the gizzard content as a percentage of the full organ, and decreased digester pH. We can see this quite nicely with this picture. Here we have the control birds and the birds fed oat hulls or sugar beet pulp. You can see very clearly the change in the size of the gizzard in response to those 2 particular fibre sources. In this study, the digestibility or apparent digestibility of neutral detergent fibre, acid detergent fibre, and acid detergent lignin was also assessed. There's significant improvement in the digestibility of those fibre fractions with the addition of oat hulls. No significant difference between the control or sugar beet pulp. And that really exemplifies the grinding action of the gizzard and being able to make more utilisation out of this less digestible source of fibre in oat hulls. Therefore, we know that the digestibility of dietary fibre is more variable and lower than that of other nutrients. The digestibility in the feedstuff, and therefore in the diet, is negatively affected by, first, the content of dietary fibre, and second, the source and hence the types of dietary fibre. In this way, digestible and metabolizable energy contents are negatively affected. There's also linear decreases in the illegal digestibility of dry matter and crude protein, or amino acids, with increased levels of dietary fibre. However, the extent of the anti-nutritive effects depends on many factors, such as the age and physiological stage of the animal, the type of feedstuff, ingredient diet processing, and of course, enzyme addition. And this study, these pictures show the impacts of dietary fibre on energy and nutrient content and availability in pigs. The diagrams here on the left-hand side show the relationship between the digestible energy content. This is nursery pigs fed different types of wheat-based diets from Western Australia. You can see the negative relationships, whether expressed as NDF, total non-starch polysaccharides, or xylose, with digestible energy content. But total dietary fibre content has impacts on other nutrients, such as phosphorus. So this is the relationship between dietary NDF on the x-axis and the apparent total tract digestibility of phosphorus. We can see an inflection point at about 15 to 17% dietary NDF, such that the digestibility of phosphorus decreases. But again, that underscores the importance of thinking about enzyme addition to diets with reasonable levels of dietary fibre in them. Of course, xylanase can be used to obliterate or mitigate some of the impacts of dietary fibre and improve digestible energy content. This is a study looking at 3 different varieties of wheat from Western Australia, without xylanase in the purple bars and with xylanase in the green bars. You can see that for 2 out of the 3 wheats, there was a 2 to 4% improvement in digestible energy content with the addition of xylanase. In this variety, Arano, in fact, the reverse occurred. There was a decrease in digestible energy content. But again, for situations where where the variety is unknown, then in general, you would want to be looking at the addition of xylanase in wheat-based diets for young pigs. And this graph shows the impact of viscosity on the true ileal digestible amino acid content in ileally cannulated growing pigs. You can see here that as the viscosity increases, this is Australian barley. As the viscosity increases, then the true ileal digestibility of lysine and threonine decreases, albeit with different slopes, reflecting the different degree of losses of these amino acids from the mucosa. And we have an interest in the use of dietary fibre to breeding sows as well. Traditionally, the interest in these fibre sources was largely related to their availability as a cheaper ingredient source. But in the last 15 to 20 years, animal welfare issues, housing regulations have changed. And therefore, in addition to a supply of energy and nutrients, fibre sources fed to pregnant and lactating sows can have broader effects, such as on satiety, constipation, and piglet factors. And this study looked at the impact of the use of a high fibre treatment or high fibre supplement to sows fed in late gestation. Very simple study, a control or a treatment group. And this was a fibre supplement with wheat, sugar beet pulp, sunflower seed, and soybean hulls, about 150 grams per kilogram of crude fibre. We can see that the addition of this treatment as a supplement, as a top dress, reduced stillborn piglets and reduced overall mortality. And if we drill down into this further and look at the cause of piglet mortality in lactation, we can see that in those litters that where the sows were supplemented with the high fibre top-up, the piglets, there was a significant reduction in poor viable piglets at birth and a significant reduction in deaths with piglets succumbing to diarrhea. So some positive effects there of adding fibre to late gestation diets for sows. And of course, dietary fibre is much talked about and much referenced with respect to post-weaning diarrhea in piglets. It is one of the many numerous strategies and additives available to mitigate post-weaning diarrhea, particularly in the absence of antimicrobial compounds. In fact, the effects of adding insoluble or less fermentable fibre to reduce post-weaning diarrhea have been recognised for many years, going back 55 years in some instances, where barley hulls or high levels of crude fibre reduced E. coli proliferation and reduced diarrhea in the post-weaning period. We conducted some studies looking at the increasing the level of insoluble non-starch polysaccharide content in E. coli challenged piglets in the post-weaning period. This is data looking at the impact of increasing the insoluble NSP content on average daily gain on this axis, and on the shedding of fecal beta hemolytic E. coli on this y-axis. We see an increase in average daily gain with more insoluble content, and a quadratic decrease in the shedding of E. coli with higher levels of insoluble NSP. This has been shown to relate also to changes in families of bacteria. So this looks at the relationship between the insoluble NSP content and the tax abundance of Christensenolaceae and Lactobacilliaceae, two families in the faeces of these piglets. We see a significant linear improvement in Christensenolaceae. These are obligate fibre fermenters and are particularly known for their ability to produce butyrate. And interestingly, we saw a decrease in Lactobacilliaceae. Dietary fibres can also have an impact in suckling piglets. So this was a study that looked at 4 different diets fed as a creep feed to suckling piglets from day 7 to day 22, while still having access to milk from their mothers. 4 diets: control diet, a diet with 1.3% alfalfa added to the basal diet, a diet with about 3% wheat bran added to the basal diet, and a diet with 1% pure cellulose added to the basal diet. These were all about the same levels of NDF and ADF. And this study looked at the relative abundance of Streptococcus suis. This is the agent of meningitis. And this looked at the relative abundance along the large bowel. in the cecum, the proximal colon and the distal colon. And there are some significant findings, for example, of adding alfalfa. Looking at alfalfa, not always statistically significant compared to other, at least in the cecum, compared to the control, but certainly when we get to the distal colon, some positive impacts of feeding alfalfa and wheat bran in reducing the reducing the relative abundance of this pathogen for piglets in the pre and post-weaning period. My final aspect today is to look at some functional fibre fractions and their impacts in pigs. We know that fibre degradation products such as xylo-oligosaccharides or ZOS, these are chains of xylose molecules linked by beta-1,4 bonds. The xylo-oligosaccharides can be produced enzymatically by the hydrolysis of arabinose islands in the appropriate biomass. And that can occur either endogenously via the addition of xylanases to the diet, for example, or from the hydrolysis of that biomass under manufacturing conditions, and that product can then be added to a diet. Both in vitro and in vivo, and in a variety of species, including man and including rats, the xylo-oligosaccharides have proven to have significant beneficial physiological and microbiological effects in the gastrointestinal tract, and this is the concept of the prebiotic, that certain xylo-oligosaccharides have prebiotic effects to benefit intestinal health, structure, and function. So, for example, in healthy humans, a study was performed comparing xylo-oligosaccharides from wheat arabinose islands or the same product with added inulin, a fructan polysaccharide. And in this study, ZOS alone increased fecal concentrations of bifidobacterium and butyrate. It increased activities of these bacterial enzymes and decreased concentrations of cresol. And cresol is a biogenic amine associated with inferior intestinal health. There's been some recent studies in piglets, and I just wish to go through those with you now. So the first study looked at feeding a xylo-oligosaccharide product as a prebiotic to demonstrate prebiotic effects, positive prebiotic effects on performance, physiology, and aspects of the microbiota after weaning. There were 3 treatment groups. Pigs were fed either a control basal diet, the basal diet with 500 milligrams per kilogram of ZOS, or a positive control, which was basal diet with 100 milligrams per kilogram of chlortetracycline. The xylo-oligosaccharide product was manufactured from corn cobs, 95% purity, a degree of polymerization of 2 to 7, and it was formed by xylose residues linked through those beta-1,4 linkages. Pigs were fed for 28 days, and then a subsample was euthanized for sample collection and subsequent analyses. If we look first at the production parameters, here we have body weight, average daily gain, average daily feed intake, and feed to gain. So really 2 points of notice here. The first is that for any of these particular measures of production, the addition of the ZOS at 500 milligrams per kilogram did not cause inferior performance relative to the positive control in chlortetracycline. And the second point of note is that the addition of ZOS in some cases, for example, in body weight, average daily gain, and feed to gain, actually improved the performance relative to the control diet, and certainly was no worse than the antibiotic chlortetracycline. Short-chain fatty acids were measured in this experiment. We have total short-chain fatty acids, and then the concentrations of acetate, propionate, and butyrate. We see here that there was a significant difference between the addition of ZOS and the control pigs, the production of much more short-chain fatty acids with the addition of the prebiotic, but it's really the concentration of butyrate that is of significance here. We see that the use of the xylo-oligosaccharide increased butyrate concentration relative to both the control pigs and the chlortetracycline fed pigs. This also translated into changes in major cecal bacterial genera. Here we have lactobacillus. The use of the xylo-oligosaccharide significantly increased the copy number of lactobacillus relative to the other two treatments, but also decreased, in accordance with the use of the antibiotic, the copy numbers of these potential pathogens in the gastrointestinal tract of young pigs. This study looked at the impact of feeding ZOS in a study with lipopolysaccharide injection, or LPS, in post-wean pigs, where measures of mucosal barrier function, inflammation, histology, enzyme activity, and the microbiota were made. This particular experiment looked at the impact just focusing on one aspect of this experiment on barrier function. This is Claudin-1. This is one of the tight junction proteins that is responsible for maintaining permeability, maintaining tight junction structure in the post-weaning period. Here we have different diets: the control diet and xylo-oligosaccharide. This was the placebo. And then the control diets, the control pigs that were given LPS, and the ZOS-fed pigs also injected with LPS in both the jejunum and the ileum. And just to summarise the major effects here, we see that in the jejunum, there was no impact of LPS on Claudin production, but the use of xylo-oligosaccharide increased the expression of that particular tight junction protein. If we look at the ileum, there was an interaction such that with the ZOS-fed, the ZOS-fed pigs that were given LPS showed superior barrier function, if you like, relative to the control pigs that were injected with LPS, again demonstrating some functional effects of xylo-oligosaccharides on barrier function. And at the genus level in this study, the use of ZOS enriched the relative abundance of so-called beneficial bacteria in the gut, such as Faecolibacterium, Lactobacillus, and Prevotella. Finally, I just want to look at the impacts of xylanase on the functionality of dietary fibre, again in the context of xylo-oligosaccharides. We know that xylanase, these are glycosidases, and this subject will be talked about further in today's conference, that catalyse those xylosidic linkages in the xylan chain. There are important benefits of xylanase, and these are well known. For example, better nutrient digestibility, growth performance, reduced variability in energy and nutrient availability, and lower diet costs. These are well-established principles with the use of xylanases. But the use of xylanase in pig and poultry diets is now going to go beyond the mitigation of just these anti-nutritional factors. There are benefits beyond that, and they may play roles in modulating intestinal health after weaning, possibly through these prebiotic effects via the production of the short-chain oligosaccharides, such as xylo-oligosaccharides. And this is just a study that is currently in press. It's been approved for publication in the Journal of Animal Science. This was a study that looked at the impact of an endoxylanase, an endo-beta-1,4 xylanase, on various physiological parameters in nursery pigs fed corn, soy, DDGS diets. The diagram on the left-hand side shows the relationship between xylanase and xylanase units per kilogram and viscosity on the y-axis. We can see that there is a linear decline in viscosity as xylanase concentration in the diet increases, but a significant impact as a main effect, or as a contrast rather, between no xylanase and all the xylanases. The graph on the right-hand side shows the relationship between xylanase. Now, this is expressed as xylanase units per day, so it's the intake of xylanase units against the concentration of MDA, or malondialdehyde. And this is a marker of oxidative stress in the small intestine. See here that as xylanase intake increases, then the concentrations of MDA decrease and they reach a threshold. Again, showing some benefits there in terms of intestinal health. So in conclusion, dietary fibre can have profound impacts on utilisation of feedstuffs. Dietary fibre is generally regarded as anti-nutritive, but the negative impacts can be alleviated, for example, by the use of enzymes. There's increasing emphasis on the functional or the non-nutritive aspects of dietary fibre to benefit intestinal health, welfare, and environmental aspects in birds and chickens. And in this respect, xylo-oligosaccharides would appear to confer some physiological benefits in monogastric animals, typically at lower doses than prebiotics. And this is a very exciting part of future research, but the attention needs to be paid to the purity of the ZOS, the degree of polymerization, and the type of substitution present in the product. Thank you very much.
Grow and Act.